Hello and welcome to Nikolai's genetics lessons, and today's question is: what charge would you expect on alanine when placed in a solution with pH of 1? And here is a variance of the answers. Now take a look at this pH scale and you would see that we can measure it between 0 and 14. Those which closer to maximum would be solutions which are basic. Those which are closer to 0 would be acidic, and the closer to medium we would see neutral. So, for example, our blood is very close to being neutral. It is about 7.4 on the pH scale. What pH scale tells us that here we can find a lot of free hydrogens, and the more free hydrogens we will find, the more acidic such solution is going to be. And the more we will find OH, free OH in our solution, the more basic our solution is going to be. For example, those which would be close to 14 would be very basic. Those which would be very close to 0 would be very acidic. At the neutral, we will find about the same number of OH groups and hydrogens. We usually don't see many OH and free hydrogens together because they just would form H2O, or molecule of water. How to memorize that basic liquids is closer to 14 and acidic is closer to 0? Just take a look at all these examples here and you would see that the heaviest is going to be a battery. And probably everyone knows that battery contains acids inside and battery is most heaviest, so would be closer to 0. So this is just a mnemonic technique how to memorize that acids are closer to 0 and basic liquids and substances are more close to maximum of the scale, which is 14. Another thing that you probably have to know, that for example between pH 7 and pH 6, the difference in number of hydrogens would be tenfold, and between 6 and 5 again would be tenfold. That means that between pH 7 and pH 5 the difference in number of free hydrogen would be 100-fold, and if you'll compare with pH 4 would be 1000-fold, and so on. And the same is true for this part of the scale in regard of the OH group. Now let's take a look at the structure of the amino acid. All amino acids has the same structure. So we have central alpha carbon, carboxylic group, hydrogen group, amino group, and R group. So all these three groups are going to be the same in all amino acid, and R group are going to be different and unique for each amino acid. Now take a look at low pH. You know that we are going to have a lot of free hydrogens, and at high pH we are going to have a lot of OH groups here. And what's going to happen to amino acid? So here amino acid is shown at the pH 7, close to physiological pH. We see that amino group here has positive charge and carboxylic group has negative charge. That's why we call this zwitterion. This means that this molecule has both positive and negative charge, which cancel each other, and we consider zwitterion to be neutral. At the pH 7, only 3 amino acid has positive charge or would be basic, and only 2 that has negative charge, or we call them acidic amino acids. Now let's read our question again. What charge would you expect on the alanine? So alanine, here is the formula of this amino acid, and we see amino group, carboxylic group, and here is going to be R chain or R group. And we see that it is non-charged. that this is non-polar and it's not reactive. All R groups that consist of the carbon and hydrogen only, we say non-polar and non-charged. That means that when we change pH of our solution, R group is not going to be affected. It's only going to affect amino group and carboxylic acid group. At the low pH of 1, we are going to have a lot of hydrogens, and let's think what's going to happen here. Negatively charged oxygen in the carboxylic group is going to attract this extra hydrogen, and molecule is going to lose this negative charge here, but the presence of the hydrogen is not going to affect amino group, because nitrogen normally has 3 covalent bonds, but sometimes also can have 4 covalent bonds, and we see here that this nitrogen here has positive charge and has 4 covalent bonds, and it cannot react with another hydrogen. It doesn't matter how many hydrogens are present here. So as you see on the overall, we are going to get a molecule with positive charge, and this is answer A. What's the importance of this information? Imagine that we are using gel electrophoresis to separate our proteins or amino acids, and by changing pH of our solution, we would be able to separate amino acids. Those which are positively charged, when we apply electric current, would move in the direction of the negative charge, and those which are going to be negatively charged would move in the direction of the positive charge. And this is all for today. Thank you for your attention, subscribe, and see you in the next video. Goodbye.